Welcome to the 9P Family webinar. This is first. I think that in the past it's been done, but this is a first for me. There will be several sessions in today's 1 hour. So thanks to Tisha Turner, the first 15 minutes. And then after that, we go to Patricia Dixon. Tisha, you'll present the entire group, huh? And then Monica afterward. Just a quick reminder, the session is recorded. At the end, we will make sure that we, eliminate the name so that you have the answers without the name of the person that responded or asked the question. So feel free. There will be at the bottom of the application, you could see a Q&A. If it's not there, go to more, and then you'll see a Q&A, and then you can type your Q&A. Tisha will remind us on how to proceed. That's about it. So if you could save the little question for the last 15 minutes, and then we can start now. Tichelle, this is to you.
Thank you. Thank you for the introduction, and thanks everyone for the opportunity to come and present to all of you tonight. Hopefully you can see my slides. So this is our first family webinar under the 9P Arch network, which I'll be talking about tonight. So I will be first talking about the genomics of 9P related syndromes, and then Dr. Patricia Dixon will be talking about genotype-phenotype connections in 9P. And then Monica Sentmanat will be talking about cellular modeling. And so that will be something I think most people have not seen before. And so I hope that will be very exciting for all of you and will be sort of the three pillars of the 9P Arch network kind of research that we're doing. So let me just go ahead and jump right in. So I will be talking about one of our most recent studies, which is called Whole Genome Sequencing Reveals Individual and Cohort Level Insights into Chromosome 9P Syndromes. So as a reminder, 9P related syndromes have to do with the P arm of chromosome 9. So here's a schematic of chromosome 9. This part here is the P arm. This part over here is called the Q arm. And so in 9P related syndromes, we're looking at things like deletion or a loss of a piece of DNA on 9P, or things like duplications, which are gains of DNA on the P arm of chromosome 9. So when we first started working on 9P related syndromes at WashU a few years ago, we started to ascertain all of the data from the literature. And when we did that, we found that when we look at the P arm, which is shown again here, this part over here is the end called the telomere. This part is the middle, it's called the centromere of the chromosome. And all of the little red bars are deletions observed in individuals with 9P related syndromes. And all of the blue are duplications. And as you can see, each of them looks quite different. And so that was our first look at 9P related syndromes. And from there, we teamed up with the Chromosome 9P Network, your family network. And your family network is so amazing. You started with one family back in 1984, and now there are over 800 families in the network and from all over the world. So in blue are all of the countries where individuals are in the Chromosome 9P network. So this was the first big study that we did on 9P, and we looked at karyotype level data. So that's a little bit low resolution, but we had it on about 700 individuals. So we were able to look at what are the common breakpoints that we see in 9P. And first we saw that if we look here at the P arm again, the telomere here, and all the way across to the middle, the centromere, we can count where the deletion breakpoints occur. And we found from this that the most common deletions are in this part of the chromosome. The second most common are in this part. And that was helpful. But from there, we decided that we needed to do more. And so we decided that in parallel to the family network, we would create a research network, which we call 9P Arch. And we call it 9P Arch partly because we're in St. Louis, Missouri, and so we have the Gateway Arch in our city, but that stands for Advanced Research in Chromosomal Health, Genomic, Phenotypic, and Functional Aspects of 9p-related Syndromes. So in this, we're interested in the genomics, the phenotypes, and the function, which are three areas, the three talks for tonight's webinar. Our 9P network is not as big as yours on the research side. We're working on it, but we have labs now and clinicians represented in the US and in Canada, and most recently in Belgium. So we are not quite fully global yet, but we are working on engaging with lots of researchers and clinicians around the world. So in our first study as a network, we were focused on the genome. And why are we interested in the genome? Well, first, we want to understand what are the critical genes, which are important for the individual and are also important at the level of the group. And ultimately, to provide a necessary framework for therapeutics in individuals who may be interested in therapeutics in the future. So the study that I'm going to talk about today was published just a few weeks ago in Genome Medicine, and there's a QR code at the top if you'd like to read more. But in this study, the first thing we did was to assemble our research network. The second thing we did was the largest ever study of whole genome sequencing of 100 individuals with 9p-related syndromes and their family members. From this, we could create a map of 9p-related syndromes. Then, because many of the individuals in our study had 9p deletion syndrome, we were able to look at several different aspects of 9p deletion syndromes. And since I'm going to go through them in detail in just a moment here, I just show them on the slide briefly here. So the 9p ARCH cohort study design looks like this, and many of you may have participated in the study and may be familiar with this already. But essentially, it starts with someone who signs a consent form and is in contact with the clinical research team led by Dr. Dixon. And Dr. Dixon's team does extensive work, medical record checks, as well as surveys, but she will be talking about that part in her part of the webinar. So I'm just keeping it brief in my part of the talk here. But if the participant is a baby or they're not a baby, there will be a determination on how much of blood that will be collected. This is done by shipping the kit to the family, and there are several ways. At least classically, we had someone go to the house for a blood draw, or the family would go to the doctor. That may slightly change in the future, but at least that's how it was done in the past. And then the blood kit is shipped to WashU, to the McDonald Genome Institute, which is one of the premier institutes for genomics in the whole world. Once it arrives here at the McDonald Genome Institute, then karyotyping goes on for each individual, a whole series of genomics, which is what my team leads. And then another part of the collected sample goes to the cell team, which is led by Dr. Kui and Dr. Sentmanat, who is on the call tonight, and that will be in part of the third talk tonight. So that's just the overview of the study. So specifically, the individuals who were sequenced in the 9p ARCH cohort, there were 100. Of those, 85 were unrelated probands, so that means unrelated individuals with 9p. There were 6 moms, 5 dads, 2 siblings who also had a 9p related syndrome, 1 grandfather, and 1 grandmother. And from this, we were able to create a computational strategy to use the genomic information from this whole genome sequencing data to build a genomic map. And what I want you to see here, this is a map that's built using statistics, and each dot in the map is an individual. And what you can see from this map is a little schematic of the P arm of chromosome 9. Here is the telomere, here's the centromere, just to reorient you. Orange means a deletion, blue means duplication. And what we can see is what we saw a little bit earlier, that people have different deletions, but now we can quantify what this looks like. So in the very middle are individuals who do not have 9p related syndromes, and we use that to set a baseline. And then from there, we see that individuals with 9p related syndromes that have deletions either have small ones near the end of the chromosome, or as we move up and to the left, they have slightly larger ones that go about halfway in. There's one individual who has a deletion near the middle. There are three individuals who are not related to one another, but have a deletion followed by a duplication. And then going from this part all the way to the right, we have duplications that are getting bigger in size. So for the first time on one map, we can see all of the individuals in the cohort. And why we were interested in this was because we understand that most of the events are very different, but we can identify some people that may be closer together in the map, and that maybe we can match those individuals to as families to talk with one another. We've thought about this extensively, and we're still in conversations about how we might be able to do this. But you could imagine these two individuals may share more in common than these two individuals. And so we think this is a useful strategy. Now I'm going to focus on 9p deletion syndrome exclusively, because as I mentioned, we had the most individuals with that in the first study. So in my lab, we're really interested in something we call precision genomics, and it's really because of the 9p network and the research on 9p that my lab actually ended up defining precision genomics because it was required in one of our papers. So 9p has helped to set my career in interesting ways. Both professionally and personally, I've learned so much. But that's just a side note. But anyway, so determining all possible relevant genomic variation within an individual to the precise nucleotide is how we define precision genomics. And so we asked, with this genomic technology, how good can we do at getting all of the breakpoints? And in fact, we can get 86.4% of all breakpoints to the precise nucleotide. This is actually much better than anyone had anticipated, and I'm very enthusiastic about what that means for the future in understanding 9p. We also predicted translocation. So that's when one chromosome moves to chromosome 9. And all of our predictions were correct by a technique called FISH. And I'll just show you an example of that. So this is an individual who has a chromosome 9 where there's a piece of chromosome 4 that has attached to it. And here are all the chromosomes in that individual. And then here they are lit up so that you can see them nicely. And the little red dots are pieces of chromosome 4. So let me just zoom in. So here is chromosome 4 over here. I'll actually go one more to make it easier. Here's chromosome 4. Here's chromosome 4. Here's chromosome 9. And here's a chromosome 9 that has a deletion as well. The little red dots only light up on sequences that are on chromosome 4. And as you can see, here we have that on chromosome 4, as we expect. We have it here on chromosome 4, as we expect. And then we have a little piece of chromosome 4 that is on chromosome 9. And so we actually can do this now for all the genomes. And this is really, really good because it helps us to get right down to the precise breakpoints. We also solved a very complex case, which I won't go into because we don't have enough time tonight, but you can check out the paper and it's one of the figures in there. And just to show you that we're doing a really good job so that, you can be confident in us that we're doing a good job. So this is the full cohort, and at the top are the reference chromosome 9s, and then each of these chromosome 9s represents an individual in the cohort. So red is a deletion, blue is a duplication, purple is translocation. It's sort of like what we saw before. There are some similarities, but many differences. So then we were thinking about this question of how does 9p deletion syndrome occur? Like, what is the mechanism? And so what we did was we took all of the deletions, and some of the people with deletions also have duplications, so those are shown in blue, but in orange are the deletions. And we looked at where on the P arm of chromosome 9, here's the telomere, here's the centromere, where did those occur? And it turned out many of the breakpoints resided in these two regions here, which we call late replicating region 1 and late replicating region 2. And what that tells us is that breakpoints are occurring due to issues with DNA repair. And so it's really a part of the chromosome that's somewhat susceptible to breaks. And so it's just by chance that this is happening. And now we have the first insights into this kind of mechanism. And here I quantified that. So if you look here, we would expect that about 17% of the breakpoints would occur in that first region, but 46 of them do, which is significantly more. And here we would only expect 6%, but we see 20%, which is significantly more. And so this is the first real insight into the mechanism of 9p related syndromes from a genomic perspective. Then we wanted to know what were the genes that were important. So we compared individuals with 9p syndrome to a control cohort. So basically individuals who don't have 9p related syndromes. And we looked at the 488 genes on 9p, and 287 of them were statistically significantly different between the two groups. That's way too many genes, right? And so what we did was we leveraged a lot of human genetic work to narrow the list down. So we used information on genomic information from the population, from things called constraint and dosage sensitivity. And we also looked at over 50,000 individuals with related phenotypes to 9p. And at the end, we found 28 genes of interest, of which 24 are deleted in many individuals in the cohort. And these are the genes. We think these are the best candidate genes right now for functional follow-up. And we have done some of that. I just put this slide here as a note if you want to check out the paper. There's a lot of detail here, but we are starting to do function on these genes. But interestingly, two of the genes had a role in mitochondria, and mitochondria are the little powerhouses of our cells. And because we had whole genome sequencing data, we could look at the mitochondrial genome in individuals with 9p and compare them to age-matched controls. And when we did that, we found individuals with 9p deletion syndrome tend to have a higher mitochondrial genome copy number, and it's significantly so. We do not know the reason for that yet, but it is a really interesting insight. And when we take the 24 genes that we've identified, and in each column is an individual, orange means there's a deletion, we find that 83% of individuals have a gene in this list that is deleted. And we think that that is really important and relevant. So we think that we've got insights into the majority of 9p. And we have much work still to do on the remainder, but this is a really big gain in discovery. So as outlined, we assembled this research network. We did sequencing of 100 individuals. And for 9p deletion syndrome, we learned things about the mechanism, achieved precision genomics, and have identified several important genes for this syndrome. In the future of this part, we would like to collect additional phenotype data, and Dr. Dixon will be talking a lot more about that. We would like to sequence the 50 individuals that we've recruited but not yet sequenced. We would like to collect more individuals to sequence and start functionally testing the genes, which Dr. Sentmanat will be talking about in a bit. So I would just like to acknowledge the members of my team who are shown in the pictures on the left who participated, and then all of my colleagues here at WashU who are incredibly important in working on this together. It's really a collaborative effort. Everyone in 9P Arch who's around the world, and all of our funding that we have had to date. And I will go ahead and turn it over now to Dr. Dixon.
Thank you, Tishelle. Let me share my screen. Okay. So thank you so much for setting this up, Dr. Turner. We're going to talk now about some of the work that was done to look at the phenotype of 9p minus syndrome. And see if I can get this to advance. Here we go. So first of all, what is a phenotype? So when we just talked a lot about genotype, which is what our genes tell us is our pattern. But the phenotype is when we look at what are the effects of those genetic changes on the body. So this is the pattern of the characteristics of the person who has a given genotype. Would be when we look for genotype-phenotype correlations, that's that phenotype. So what is the phenotype of 9p minus syndrome? How can we refine our understanding of it? And can we untangle the contributions of each 9p genotype to that phenotype? So this work was published in 2024. It was done mainly by Rodrigo Starasta, who graduated from our group and is now a geneticist at Oregon Health Sciences University. So he took the first 49 probands, or people with 9p minus syndrome, and these people had had a confirmation of their genotype by a chromosomal microarray. And then they underwent the whole genome sequencing in Dr. Turner's lab to evaluate that 9p region for breakpoint definition. We had a REDCap extraction tool. REDCap is just a computer database that we use with studies to put data into, and reviewed medical charts. And Dr. Starasta conducted a phone interview after chart review to fill in any missing details. He then aggregated the detailed phenotypes into broad terms. I'll kind of show you how that went. So part of the paper is simply cataloging the different phenotypes within the cohort. And so these next few slides just go through some of the statistics that we reported. First of all, neurodevelopmental. We found either developmental delay or intellectual disability in 98% of the cohort. There was low muscle tone or hypotonia in 87%, autism spectrum disorder in 53%. Attention deficit or hyperactivity in 43%, anxiety in 20%, tics in 13%, and major depressive disorder in 9%. Seizures occurred in 28.5%. And if you had a brain MRI, about 26.9 or 27% of the cohort had abnormalities of the corpus callosum, which is a structure that connects the left hemisphere to the right hemisphere. Corpus callosum abnormalities, interestingly, even though it's a very large structure, most of those have no clinical impact on your functioning. Phenotype, the immune system, we saw allergic disorders in 59%, including eczema, asthma, and other allergies. Frequent infections in 28%. 54% reported vision problems, 43% the trigonocephaly, or the which is that craniosynostosis where there's a ridge in the center of the forehead. Strabismus, 43%, sleep apnea in about a third, hearing loss in 19%, clefting, cleft lip or palate, which is what that refers to, in 18%, and tear duct obstruction in 15%. About 36% had low blood sugar as newborns. 15% had low thyroid or hypothyroidism, and almost 10% had growth hormone deficiency, which is typically going to be associated with short stature. Congenital heart defects were seen in 43%, most commonly something that's very mild called a patent foramen ovale, which is a normal hole that you're present in the womb, allowing the blood to shift from the left to the right, right to the left side of the heart, and that's supposed to close over at birth. And so if that doesn't, it's called a patent foramen ovale. Gastrointestinal problems such as constipation was very common. GE reflux or heartburn in 52%, hernias of the belly button, 33%, pyloric stenosis in infancy, 7%. And then genital defects in 73% of XY male individuals with complete ambiguous genitalia, and 9%. Genital defects in only 22% of XX individuals, and none of them were what we call ambiguous or male appearing in female, otherwise female gendered individuals. We saw urinary tract abnormalities in 20%. Most common was something called vesicoureteral reflux, which is when the urine, instead of just going out from the bladder, sometimes refluxes back up to the kidneys. So Dr. Starasta did two approaches to evaluate genotype-phenotype associations. One is this minimally overlapping regions with the smallest deletion size that was common to individuals who had a given phenotype in common. And then another was unique non-overlapping regions, kind of marching out the different unique regions across individuals in the cohort. And I'll show you what both of those mean. So you can't read all the little writing on here. Well, maybe you can. I don't know get how close you want to get to your screen. But each of these graphs represents individuals with a given phenotype. So up here is seizures, for example. Here is ADHD. And then on the far right, I'll drawing your attention to CACUD, which is congenital anomalies of the kidney and urinary tract. And then congenital heart disease, those sorts of things. So, each of these bars represents an individual in the cohort who has this phenotype seizures and the deletion size that they have in common. So, this is from here to this point is the smallest um, the minimally overlapping region or the biggest size where that they all share in common. Over here in congenital heart disease, you can see this one individual drives that who has the smallest deletion and congenital heart defects, and everyone else also has that same region deleted. So that was that approach. And so here's a zoom in of that congenital heart defects graph showing different individuals. This is this pretty small region that they all share in common, and it contains 8 what we call OMIM genes, OMIM being the online Mendelian inheritance in man database that catalogs genes that we know what they do. And then if they have a phenotype, meaning that that gene is associated with a given disease, then it'll have an OMIM association with the disease. So these are 8 OMIM genes. And interestingly, as you can see, there's CBWD1, DOC8, DMRC1, 2, FOXD4, KENK1, and WASHC1. So this paper came out a little bit after ours with finding a chimeric KENK1 DMRT1 transcript that was associated with a congenital heart defect segregating across 5 generations. So I thought that was really interesting that that gene popped up in this paper as well as within that region that was shared by our cohort with congenital heart defects. So the next approach that Dr. Starasta undertook was this unique non-overlapping region. So if you marched out the individuals with where their deletion is mapped across 9p, so this person's deletion starts at the end and goes to here. This individual's starts here, so you draw a line there and it goes to here, draw a line here. So each of the start and end points of the deletion creates a new line, and then each of those is a new region that you can use to just map where the genes might be that you want to look at. And so using this unique non-overlapping region approach, he mapped the different phenotypes, and not all of these ended up with a hit in this cohort, but he did find a few. And for example, trigonocephaly had a big peak at a certain area, and that ended up being in where from one was. So there was one area where there was trigonocephaly and FREM1 deleted, but so 100% of the individuals with this complete area here that was deleted had trigonocephaly. This individual didn't have that region completely deleted and did not have trigonocephaly with a very high odds ratio. So we knew FREM1 was the gene associated with trigonocephaly. So this just kind of illustrated that this is an approach that might be helpful in locating genes that are associated with a given phenotype. This is just another, I'm showing that. And there's a paper that FREM1 is associated with trigonocephaly or metopic craniosynostosis. There was, interestingly, one region that trigonocephaly was seen in an individual without that FREM1 deletion. We don't know if there's a gene in here, but another paper that was published after ours in March of 2025 also thought that that same region that Dr. Starasta mapped might be a second locus for trigonocephaly. And so it's still not clear what the gene is in there, but it's interestingly that another group also found that that region was a potential area where a gene might be for trigonocephaly. And that is all. I am currently looking for another student who can pick up where Dr. Starasta left off and hopefully analyze a few more of the phenotypes now that we have a little bit more data since he had done that analysis and see if we can find some more phenotype-genotype correlations in the cohort. So with that, I'll stop sharing and I think is it Monica's turn? Okay.
Okay, I'm going to share my screen. Okay, let me make sure. Okay, yeah. Okay, so I'm going to talk about cellular modeling of 9p related syndromes using stem cells. And I want to start with giving you a sort of a sense of what makes stem cells such a powerful tool. And it really falls into two bins as to why they're so powerful. The first is that they are a unlimited resource in terms of self-renewal potential. And this is quite different from the origin of where they came from. So where they originally came from, and I'll talk you through sort of the steps, is they originally came from, in our lab, a cell that had limited self-renewal potential. So in most cases, we will get cells from patient samples that are skin punches. So we may isolate fibroblasts, and you can also isolate skin cells from, other sources, or we can isolate peripheral blood monocytes from blood samples. And all of these cell types have very limited self-renewal, meaning that after a few cell divisions, they may putter off and sort of die off. And the other thing about these cells is that their cell identity is fixed. And so what we do is we take these cells that came from patient samples and we add a cocktail of proteins that convert them into a new cell identity, and that is the stem cell. And now as a stem cell, they have unlimited self-renewal, which means that we can bank them and we can store them for years to come. And as we develop new assays and new tools, we can go back to them and use them for these different assays and workflows. The second thing that makes them really exciting and unique tools is that they are able to be pushed into a different cell identity. So from a stem cell, they can then be pushed to a disease-relevant cell type. And in particular, a neuronal cell type or a cardiac cell type or any other type of cell that is specialized and affected in that diseased individual. And this can be done in a 2D system, just in a culture dish, or you can make increasingly complex models. And these can be three-dimensional in vitro models. And as these models become increasingly complex and big, they can assemble different cell types within that structure. And that means that that diversity can again be probed, and those individual cells with these different cell types that are disease relevant can be used to get activity readouts or drug screens, and ultimately to develop new treatments that are targeted for that disease state. And so one of the things that we were particularly interested in is getting into even greater complex models. So one of the gold standards that is used for determining whether a stem cell has that property that I was mentioning, that is that it's able to turn into, under the right conditions, into just about any other cell type. It's called the pluripotency of the cell. the iPSC. And that pluripotency can be probed using something like teratomas. And that process is where the cells are injected into immunodeficient mice. And so the cells aren't rejected by the mouse. And they're just injected into the hind leg. And the cells will then develop practically all three germ layers. In fact, they will develop all three germ layers as long as they're pluripotent. And so this means that they can grow a mass of many different tissue types. And this is what is signature of truly pluripotent. And it would be the layers are the ectoderm, the mesoderm, and the endoderm. And so each one is developing different cells that are particular for that cell lineage in a single mass. So this is a very complex now mass that you can use to interrogate all these different cell types. And what makes this very exciting, and it's different from the typical organoid that I showed in the previous slide, is that this is now a vascularized tissue. And in this new microenvironment, you can not just get these different cell types, but you can get them in these very mature states. And the reason that this is particularly powerful, and especially for diseases that affect a constellation of clinical features, so things like in 9P related syndromes, you may have things like cardiac issues or sort of autistic type behavior, or maybe things that are, trigonocephaly or muscle tone, things that are of interest in but they're different cell types that are affected. And among all the different genes that are affected, it's interesting to be able in a single model to probe all these different cell types that may have different features. The other thing about teratoma is, and I won't get into the second half of it, but the first half is that you get this all-in-one, all three germ layers, but it's also a very powerful model because it's very versatile and it is sculptable. So if one were interested in just cardiac into a very well-developed, large mass of cardiac-like cell types, it is sculptable in the sense that by doing certain genetic manipulations, you can leverage those genetic switches to enrich for that lineage. So, there's a lot of flexibility within this system, but ultimately, the goal is to harvest those cells, single cell dissociate them, and probe each cell for its unique sort of cell type identity. And we do this using single cell RNA sequencing. And we're able to essentially identify the transcriptome for each one of those cells, which lets us understand what is the difference between the affected versus the control. How is the phenotype in the transcriptome being affected in the presence of these different deletions? And so in the literature, what we had seen is that we know that in wild type or in control lines, this is definitely a feasible thing to do. So previous work has shown that, tens of thousands of cells can be isolated, and you can plot the different cell lineages that are isolated from these teratomas, and they're very diverse. It's a whole host of different lineages. All three are identified in terms of ectoderm, endoderm, and mesoderm. And in fact, you can even see histology in these like beautiful stainings of things like ciliated respiratory epithelium and adipose tissue and smooth muscle if you kind of zoom in into the screen. But the thing is the literature that specifically looked at teratomas and their ability to do this had not done this using patient lines. And so what we were particularly interested in is if we use a 9P deletion line, are we able to recapitulate any of the sort of cellular phenotypes that we would expect? And so that's what I'm going to show you next is this system something that we could use to model 9P related syndrome? And so what we started with were two stem cell lines, these two iPSC lines that were derived from a patient and a father. And then we also included some control lines that had been published before, just as a sort of proof of concept that yes, we're able to recapitulate what has been published in the literature. And we also included a positive control that we knew would be absolutely defective for ectoderm lineage development. And so something that would be very low-hanging fruit that we knew, okay, we're going to have a hard time developing neuronal cells from this line. And so below that, I have an example of what a teratoma looks like. And so this is dissected from a mouse. And again, in this particular example, there's the control H1 line that was used. The stem cells had been injected into the immunocompromised mouse, and that's one of the masses that was isolated and assayed. And so here I have a graph of the pretty much just the masses. So even without going into a lot of like the really fancy omics type data sets, and just looking at the masses and the phenotypes of these different the development of these teratomas, we can see on the far left that the ASCL1 knockout, and that particular gene is critical. It's called a pioneering factor for ectoderm sort of neuronal lineages that had been knocked out in the stem cell line. And so it actually failed to form just about anything. So the mass was very low. And then next to that, we have in green the control lines, so the H1 and then the father lines. And then right of that is the patient lines. And in the patient lines, we noticed that the masses of those teratomas turned out to be actually a bit smaller than the father lines. And this was reproducible across all the teratomas that we harvested. So it suggested that at least in this sort of artificial system, there was some trouble in terms of producing all the lineages efficiently in the patient line. When we did staining for the teratomas, we observed all three germ layers: ectoderm, endoderm, and mesoderm, which is what we would expect. So they all three of them formed, and it showcases that yes, this assay is working, and definitely, at least in the control lines, we are proceeding with the method as we expected and correctly. Ultimately, what we identified, if you harvest or if you pool all the cells that we got from the samples together, both the father and the affected individual, we ultimately identified it was along the lines of 30,000 cells, and there were a total of 18 lineages identified. And so this was very exciting because this is, again, in a patient line that we weren't sure if we would be able to do, and we were clearly able to identify multiple lineages. When you separate out the ectoderm, endoderm, and mesoderm lineages, ectoderm being the more like neuronal cell types, what we identified was that in the patient samples, we observed depletion of the ectoderm compared to the father control. And so that was one observation that we came away from. And when you look at within the 9p deletion region, which Dr. Turner had shown previously, where there's like a set of various genes that are known to be affected across many individuals. In this particular individual, the bottom row shows all the various genes that were affected for him. And among them are many of the ones that Dr. Turner showed. And on the left, we have the various lineages in which we can detect one or more of these genes being expressed. And ultimately, what this really means is that across patient and father, that means for all of these genes and these different lineages, we're able to start probing what are the cell-based sort of transcriptional signatures that are different between the two, which is really exciting. And in particular, two of the genes that Dr. Turner had mentioned, which is RFX3 and PTPRD, we were able to act, and we actually have some of the highest expression across clusters. So that's both of those. And again, those are of particular interest because you do see them recurrently deleted in many of the patients. And again, consistent with what we would expect is because this is the patient only has one copy and the father has two copies of this gene reproducibly in RFX3 across all these lineages, the father has higher expression in cells compared to the son. And that is also true for PPPRD, which is what we would expect to see. And ultimately, we also see that ectoderm lineage has lower abundance of cells and also RFX3 expression in patient samples. And so this begins to open the door for us to be able to start probing what are the pathways that are perturbed in 9p neuronal cells if we were to just look at that cluster. And in total, we see that there's a lot of dysregulation, right, that we can start pinning down. We have 216 genes that are downregulated and 1,569 that are upregulated in patient versus father, and this is in early neurons. And in particular, what we found to be really interesting was that when you look at the enrichment profile for downregulated genes in the patient sample, you can start seeing these gene ontology trends emerge that are consistent with the types of expectations that you would have. So, for example, they are consistent with things like autistic behavior, seizures, intellectual disability, autosomal dominance inheritance, language impairment. So a lot of these gene networks that are dysregulated in the patient are consistent in these samples that we're collecting, which again is just to sort of show that this model is sort of working as advertised and potentially could be used to really probe in and discover new things in this system. And additionally, the downregulated genes that we identified were also involved in things like microtubule cytoskeletal regulation, neuronal projections, synaptic vesicle pathway, layer 5, like all these things that, you know, they're probably like, what does this even mean? But generally they are, the theme is that they're neuronal type genes that have been impacted and downregulated in patient samples. And so it essentially means that, again, this as a model is definitely I think a tractable system that can be used and applied to study this particular disease. And that to us is really exciting because of the power that it gives you. It's giving you in a single assay so many different types of cells, which is pretty unprecedented. And so in summary, what I've shown you is that we were able to successfully, in a single assay, produce all three lineages, and we confirmed that by staining and single-cell RNA-seq. In total, 18 lineages were identified. We show that there's global dysregulation, and it points to mechanisms involving migratory programs and excitatory inhibition function. And the data supports that we can use this system to capture cell type specific deregulation for patients with a complex disease. The caveats are that processing has to be done as teratomas develop, and they may not all be ready at the same time. But I think ultimately the next steps are expanding to other 9p samples, imaging tissue blocks to further characterize disease cell states, screens and like complementation screens, drug screens, and ultimately with the goal of uncovering cell type specific drivers of disease, their contribution to broader gene dysregulation, and how to rebound to a healthier state. And with that, I'd just like to up the list of all the folks that work on this, and in particular our director, Zhao Shui.
9P Arch Webinar, Questions and Answers. Is there a gene for hypotonia related to 9P? A clear connection has not been established yet. This will be the focus of future work with additional sequencing and a larger participant cohort. Is stem cell therapy something beneficial for people with 9P-? There are a few types of stem cell therapies. Medically, the main approach is hematopoietic stem cell transplantation, which uses donor bone marrow and is only appropriate in limited, specific cases, not including 9P- syndrome. In contrast, unproven stem cell injections are not recommended, as it is unproven and there is currently no reliable way to deliver a sufficient number of cells for that approach to be effective in 9P. Folate deficiency in 9P? Clinical data on folate deficiency in 9P is not available. The folate receptor alpha gene is not on 9P, and we have not studied it in relation to 9P. Are there other chromosome deletion studies occurring? There are studies of other deletion syndromes, such as 5P and 8P. Mitochondrial effects? Excess of mitochondrial genome. However, the molecular reason is unknown. Future functional studies can address this issue. How could we get involved in clinical trials or research? Email at 9p-at@wustl.edu for the research study. Not aware of any ongoing interventional clinical trials. Have you noticed insomnia issues? People with sleep apnea have been mentioned and lack of perception of time mentioned. How does the section of the chromosome that's missing? Page 22. Link to the gene codes. We have precise coordinates for all genes in the genome. So when we examine the chromosomal bands, we can map and intersect the genes directly with those bands. How old is our oldest participant with 9P? An audience member offered that they were 64 years old. The presenters did not have the age range at hand during the meeting. Some of us have a one-on-one with results of findings on our child. Will we get updated results for them as you have found more? We are currently working to secure additional funding for this research. If we are successful, this will be an important next step in the project. Have you been able to start to process the samples that were taken during the 9P family reunion this year? We have processed the samples to bank the cells and collect the DNA. We are currently working to secure additional funding for this research. If we are successful, full processing of the samples will be an important next step in the project.